talk about citric acid cycle, electron transport chain, all of these stuff, eventually, the reason why they're happening, right, is because we have oxidation, right, or we have oxygen. We have oxygen, right, so in an aerobic reaction, eventually we're going to take all that oxygen we have and try to yield as much ATP as possible. Yeah, I'm starting off with this. So with glycolysis, you guys know that you only yield roughly negative 84 kilojoules per mole. So it's not a lot of energy. I mean, just oxidizing glucose won't give you enough really for even basic survival. So that's why you do citric acid cycle. That's why you do the electron transport chain, because eventually you're going to yield almost negative 2,400 kilojoules per mole in terms of free energy. So that's really favorable. You like that, right? But it's also important to recognize that this whole process is called cellular respiration. I mean, you're using the oxygen, whatever. But you start out with acetyl-CoA, right? You start with acetyl-CoA, and after that, we go into the citric acid cycle. Then after that, we go into electron transport, right? So you can think about it that up until everything coming and converging into acetyl-CoA will eventually be catabolized, right? We're breaking down, okay, oxidizing it until you get more ATP and energy, okay? So from acetyl-CoA to electron transport chain, we're catabolizing, we're breaking things down, we're trying to get energy. But if we were to do something like all this energy that gets released, right, ATP, right, we can do these and do synthesis with them, right? You can take the energy that you yield from this whole process and put them towards maybe fatty acid synthesis, right? You can put it towards cholesterol synthesis, and maybe you can even do gluconeogenesis. So you see how you can catabolize acetyl-CoA, and then after you get all this energy, you can do an anabolic reaction and start creating stuff. Okay, again, that's why metabolism is so important. And you can see how acetyl-CoA is kind of like the way to really start that. Okay? I mean, as much as we talk about glucose being oxidized, acetyl-CoA is pretty important. So now you have to talk about how do you get to acetyl-CoA? How do you actually get this product, and how do you get through all of this? Well, that's where the pyruvate dehydrogenase complex comes into play. So there is a picture in slide, let me check what slide it is. It is slide 13. Slide 13. You should really, if I were you for your exam, if you can write out slide 13 and know everything that's happening in that, you will understand PVC, like 100%. I think I wrote it out maybe like 3 or 4 times, and from there, I was able to actually you'll do well on the exam. Like, that's just a fact. I know that. I know you'll do well. Especially if you're a picture, photographic memory, whatever, that will serve you right, okay? So, with PDC, okay, pyruvate dehydrogenase complex, it has 3 units to it. It has E1, it has E2, and it has E3. E1 is on the outside, right? E1. I mean, it's like on the outside of the complex. E2 is in the core of it, and E3 is on the other side of it. So, let me draw it real quickly. It's like this. E1, E2, E3. For the PDC complex, you have 5 coenzymes. We talk about NAD, FADH2, we talk about TPP, we talk about lipolysine, and we talk about CoA, right? These 3 coenzymes, at the end of the day, are going to get you the acetyl-CoA that we're talking about, okay? So let's start with E1, okay? E1 is on the outside. It's part of a protein, okay? We start out with pyruvate, right? We just finished glycolysis. We get this pyruvate, and now we are still in an environment that has a lot of oxygen in it, okay? So we want to make more energy, as much energy as we can. So the first step in doing that is to use this complex, PDC, so that eventually we can get to acetyl-CoA, which would be the starting reactants for citric acid cycle. So pyruvate is going to come in here, and it's going to do a reaction called TPP, or we use that coenzyme TPP for it. What TPP does is that you know that pyruvate is 3 carbons, right? Pyruvate is 3 carbons. The TPP is actually going to transfer 2 of those carbons. Okay, it's going to transfer 2 of those carbons and lock it in E1. So I'm going to write it right here. Carbon part. I'm not writing it all out because it would take too much time for me to do that. But again, if you go to slide 13, that's actually what I'm referring to, and I'm trying my best from my own memory to see if I can write it out, okay? So what's going to happen here is TPP is going to take 2 carbons, it's going to attach here. At the same time, you're going to get rid of carbon dioxide. In your problem set, she asks, what step in PDC, right, is going to be irreversible? It's this step right here. As soon as you can take carbon dioxide off, it is going to be very hard to put that back on. That's why we say the first step of PDC is irreversible. The rest of the steps that I'm about to show you, those are reversible. It's because they're redox reactions. Redox reactions typically can go back and forth. You can oxidize it, you can reduce it. But as soon as you have something involved with decarboxylation, something regarding carboxylation, it requires energy, okay? And when it requires energy, it gets harder. Yeah, just come in. Why not? Oh, I didn't see that. When the CO2 comes off, it becomes irreversible, okay? Anything that has redox, NADH, FADH2, those types of things, they're reversible. Unless it has to do with CO2, then I'm going to tell you that it's irreversible. According to Dr. Erb, anything involved with CO2 is going to be irreversible. Unless it doesn't, then we're fine, right? It's going to be reversible redox. So, as soon as this happens, TPP, okay? In E2, you have this thing called lipoic, I have to make sure I say it right. Lipoyl lysine, okay? Lipoyl lysine is this lysine amino acid here, okay? And it's an arm. I like to say that usually things that have lysine attached to them, like when we talk about biotin, right? With the pyruvate carboxylase in the first step of gluconeogenesis, it uses biotin, has that lysine arm like that. It's the same idea. There's a lysine arm, right? And it actually has 2 sulfur groups. And it's actually a disulfide bridge, okay? In this specific part of E2, it is fully oxidized, right? It is fully oxidized. And no, I did not correctly write the actual orientation of it. I didn't write the right carbons and stuff like that. But just the idea that you have the 2 S's there, that's important. I would write it properly, but I'm not going to at the moment. It's going to take too much time for me to count the carbons and stuff. But this is what we call acyl lipoyl lysine. Obviously acyl because of this, and then the lysine arm. Right now it's oxidized. What's going to happen here is in E2, this arm is going to take the 2 carbons that are right here, and it's going to attach. Sorry, I'm going to make it look like, I want to make it a little bit easier to understand. Okay? What's going to happen here is that the lipoyl lysine, okay, is going to grab the 2 carbons, okay, that we have after pyruvate lost carbon dioxide, and it's going to get attached to one of the S's, one of the sulfurs, and the other sulfur is going to get reduced. When we see something like this specific compound can become oxidized, half oxidized or half reduced, and then fully reduced. There's 3 orientations for it. In this orientation, you're in the halfway point. Half of it is oxidized, half of it is reduced, okay? And again, I'm going to ask you real quickly, what is the, what's the coenzyme for this? For this one right here, lipoic acid, right? I mean, right here it's lipoyl lysine, so it kind of makes sense that you call it lipoic acid. That's the coenzyme for it, okay? Now, when you're still in this subunit, you're still in E2, the next part is to add CoA, right? Because the whole objective is to get acetyl-CoA. Right now you have this acetyl group, okay? But this thing needs to come off from the lysine arm, and you need to make CoA. That's why the enzyme CoA is involved in it, okay? So what's going to happen here is that we're going to see this lysine, okay, is going to break off right here. It's going to break off, and you're actually going to get, I'm going to make sure I write this correctly. Sorry, I just want to make sure it's right. Do you see what I just did? Do you see what I did? All I did was, the CoA is going to come in and insert itself right here, break this sulfur, right? The CoA is going to come and attach here, and all of a sudden the S is going to become fully reduced. Do you see what I did? Yes or no? Say no if you don't see it. I feel like I see it. Okay, so oxidize, halfway oxidize, and then finally it's fully reduced. When this lipoyl lysine is fully reduced, I can automatically tell you that this acetyl-CoA is ready to go. That is acetyl-CoA. You just made acetyl-CoA right here. It's done, right? But now the whole thing is that it eventually goes back to the oxidized portion. So how does it get back to the oxidized portion? That's where the cofactor, or the coenzyme, FADH, comes into play. FADH2 is found in E3. So, it's a prosthetic group. When we say it's a prosthetic group, FADH2 does not come off. It sticks right here. So what's going to happen here is that you're going to get these hydrogens are going to be given to the FAD, generating FADH2, right? Because, I mean, it's two hydrogens, you give it to FAD, you get FADH2. But the thing is that this FADH2 never leaves. So, what it's going to do is that instead of FAD, or actually, you know what? Let me make it even better. Right? So, give me a second. The two hydrogens are going to come off of this thing, and you're going to give it to FADH, right? And it makes FADH2. But FADH2 is a prosthetic group. It's stuck in E3. It doesn't come off. So, what we're going to do instead is we have this thing on the outside, NAD+, right? The NAD+ is going to grab the two hydrogens, and it's going to make NADH plus H+. And guess where you can put this in? Electron transport chain. And all of a sudden, what happens to this group right here? The two sulfur groups, they're going to make your sulfide bridge again because you took off the hydrogens and they became oxidized. I just kind of drew out very quickly PDC complex. Does it make sense? What I just did?
This happens 2 times, right?
Happens how many times?
This happens 2 times?
No, it's just once.
For the 2 pyruvate?
Oh, 2 pyruvate, yeah. If you're talking about 1 pyruvate, yeah, you could do that, yeah. Because technically, pyruvate can go into anything, right? So, I mean, but in terms of oxygen, yeah, you could have both pyruvate go into this. Does that make sense? I know I just kind of wrote it out, but again, if you can see what I did and how everything is transferring itself, the process makes it easier to understand. I mean, you can't get rid of FADH2 because it's stuck there. So why don't you just be efficient and put it onto NAD+ and give it to electron transport chain, right? You make the process favorable in itself. Making energy is favorable. And then obviously you go back to oxidize so you can get the next pyruvate to come in. TPP. Everything happens again. Are you guys good? Does that make sense? I went over PDC. I think it was, I don't know. It was okay? Yeah? Yeah. It goes back down there because you took off the two hydrogens and you gave it to FAD and it becomes FADH2.
So like when it goes up there and then reduce,
Yeah.
where do you get those other?
Where do you get these hydrogens?
No, the sulfur is up there. Where do you get that?
Where do I get these sulfurs from? Well, the sulfurs are from here. The lipolysine group already has 2 sulfurs. So as soon as you transfer a group, it only hits one sulfur. So you have the acetyl group there, but this SH obviously broke a disulfide bond. So you'll have this with a hydrogen on it, right? And then after that, adding the CoA just eliminates this group. And then you're left with just two SHs again, right? If you don't have something attached, you need to make sure those carbons, or I'm sorry, those electrons are not going to be conformal charges, right? You want to keep it neutral. So leaving that hydrogen there makes it neutral. Does it make sense? Okay. 1 second. Okay. So you have to be able though to regulate PVC. When we talk about regulation, let's say for example, guys, you have a lot of energy. You know that at the end of the PVC complex, at the end of using this, right, you get acetyl-CoA, and acetyl-CoA can go to what?
Citric acid cycle.
Citric acid cycle. Citric acid cycle goes to what?
Electron transport chain.
Yes, electron transport chain. So what are you making?
ATP.
Yes. If there's too much ATP, do you think this process will be happening? No, it doesn't. High levels of ATP are going to turn off the PVC complex. So let's write it. We'll stop the process if there's increase in ATP. What else do you think can stop this process?
Low amounts of CN. Wait.
Yeah.
Low amounts of CNP means there's a high amount of ATP.
Yeah, that's true. That's true. Let me save that one for the next one. Another way of knowing that you can stop this, feedback inhibition. If I have too much product being made, if I already have a lot of acetyl-CoA, do you think it's going to continue making more? No. Okay. So acetyl-CoA, if there's too much, it will stop it. If I have a lot of NADH being made, do you think this process is going to happen? No. If I already have energy, there is no point in me making more. Now let me ask you, how do you make the process happen then?
AMP.
Yeah, AMP. So get this engraved in your head. When there is high levels of ATP, that means AMP levels are low. So what can I tell you? If AMP levels are low, then you don't do this process. But if I say AMP levels are high, that means that ATP levels are low. So what does that mean? If ATP levels are low, I'll do the PDC, right? I'll do the pyruvate dehydrogenase complex. Does that make sense? You just have to know the analogy between them or the correlation between them. Okay, obviously, I'm just going to write the rest of them because I don't think it makes sense for me to go over it. Same idea that if you have a lot of reactants, or if you have a lot of those coenzymes, if I have a lot of CoA, why don't I just continue making more acetyl-CoA, right? It can push more. If I have a lot of NAD+, this can push to make more NADH, right? I mean, this makes sense. Does that make sense? The idea that you're having these so-called like reactants to make more product, same idea. Le Chatelier school. If I have a lot of reactants, I'm going to go ahead and make a lot of products. Okay?
Does phosphorylation inhibit E1 or
Yeah.
does it activate E1?
It does. So now let's talk about this, right? We have an issue though. When there's a lot of ATP, what's going to happen here is that, right, PDC gets stopped. But the way it actually stops is through phosphorylation. When you have high levels of ATP, what's going to happen here is that they're going to phosphorylate E1. When you phosphorylate E1, you stop this whole process. So it's actually done by a kinase, which makes sense, a kinase. Remember what a kinase is? A kinase is something that's transferring a phosphate group from a very high energy carrier. Okay, if you have a lot of ATP, you're going to use a kinase on this, taking a phosphate off from this and putting it on here, and it stops PDC. Did that make sense what I just said? Did it make sense? Say no if it didn't. Just say no, I don't get it. No, that's fine. So, if I have high levels of ATP, I know I'll shut down PDC. But the way to shut down PDC is through a kinase. What's happening here is that you have so much ATP. The kinase is going to come in right? Because kinases transfer phosphates from high energy carriers. The high energy carrier is ATP. It's going to take the phosphate and put it onto E1. As soon as you put the phosphate onto E1, you've eliminated PDC, and that's just because you have high levels of ATP. Are you good? Yep? Okay. Next thing I'm going to talk about, okay, first of all,
Oh,
good.
I was just wondering, do we need to know what specifically phosphate binds to, like if it's the
No,
active
just
site or-
phosphor. No, just that it's on E1. Yeah, just that it's E1 and it's kinase.
It's,
Okay, I'm gonna erase this.
It's allosteric. It's not the active site. Yeah.
So let's erase this. One thing, guys, that I noticed is that people think that the citric acid cycle is really hard. It's actually really not. It's the same idea. Glycolysis is actually pretty easy too. Now, I really don't think there's much to talk about the citric acid cycle. Like, know the enzymes, know the structures, and remember how it works. Like, I can't even be that much more, I guess, like, what's that word? That's like concise about it, right? So, I will though talk about 3 of the main points of regulation. The main points of regulation in the citric acid cycle are citrate synthase, step 1, step 3, isocitrate dehydrogenase, and step 4, alpha ketoglutarate dehydrogenase complex. So I feel like it might sound like a lot, but it's not a lot, like 100%. All of these cycles have the answers in it. You just have to look at the image. It is telling you what you're doing. Every enzyme you see is telling you what you're doing. If I see, give you an example, if I see citrate synthase, I know for a fact that I'm taking oxaloacetate, 4 carbons, and I'm mixing that with acetyl-CoA, 2 carbons, to make a 6-carbon compound called citrate. And it's called citrate synthase. Like, it's really like, that's it. But with this enzyme, specifically citrate synthase, okay, with citrate synthase, it has a certain conformation to it, meaning that you have to bind everything correctly before you can start the citric acid cycle. For starters, you know that it is oxaloacetate plus, right, acetyl-CoA, which makes citrate, right? And that's that enzyme, right? The thing is, though, is that this enzyme, okay, you have to properly bind it first. Meaning that if this is my enzyme, citrate synthase, we'll call it CS for short. You need to bind oxaloacetate first. I'll call it O. You need to bind oxaloacetate first because once you bind it, it makes a little opening site for acetyl-CoA to come in. As soon as acetyl-CoA comes in, boom, I can get citrate. But you have to bind oxaloacetate first. The problem here is that people think, oh, if I just bind acetyl-CoA first, I'll get citrate. You're not going to. You're not going to, because the problem here is that oxaloacetate is the one that makes the opening for acetyl-CoA. It makes it. If I don't put oxaloacetate in it, acetyl-CoA can't come in. That's just a fact. There is no place for it open. If it does though, right, then all of a sudden this will open up. The whole thing is you have to bind this first, really. Okay? Alright. That's that. Let's see if I'm hitting anything else. Okay. Isocitrate dehydrogenase. Can anyone tell me what's important about this enzyme? Isocitrate dehydrogenase. Can you tell me something important about it? Just one thing, at least one thing. Okay, I'll tell you then. Isocitrate dehydrogenase, step 3. And we're gonna call it, oh god, isocitrate dehydrogenase. There's no complex. It doesn't say isocitrate dehydrogenase complex. It doesn't. It just says dehydrogenase, okay? And I'm gonna explain why in a second. Isocitrate dehydrogenase is only important because it is the first step where you lose carbon dioxide, right? The first CO2 loss. Remember when I told you that when you lose CO2, it's an irreversible process? Yeah, this is an irreversible process, okay? It's irreversible. Notice that in all the metabolic processes we've talked about, those irreversible steps are usually the steps that are the main regulation points. And it's irreversible, so I can definitely tell you that it is a main point of regulation, all right? It's irreversible. Now, what else did I want to say about it? Oh yeah, you also make NADH, okay? You also make NADH. It's your first NADH that you make actually, right? So that's your first NADH. That's what I wanted to say, okay? That CO2 loss, irreversible, NADH. Okay, now step 4 is a little bit interesting, okay? Same idea, but instead, what it is called, it's called alpha-ketoglutarate dehydrogenase complex. That's right. So, okay, I am emphasizing that it says complex because this enzyme works very similarly to the PDC, Pyruvate Dehydrogenase Complex. This is just alpha-ketoglutarate dehydrogenase complex. So, think about it. This is actually pretty cool in my opinion, okay? What does this reaction catalyze? Or what does this enzyme catalyze? What reaction is it catalyzing? It is catalyzing alpha-ketoglutarate, and it is making succinyl-CoA. Okay, first of all, remember how pyruvate makes acetyl-CoA? Okay? In this, you're making succinyl-CoA. This complex works very similar to PDC because you use TPP, you use that lysine arm, okay? But more importantly, you also get CoA, right? The idea that you're attaching CoA, the same way you had acetyl-CoA, you're putting a CoA and making it succinyl-CoA. And then obviously you make NADH, right? And then obviously you lose CO2. Where do you think you lose the CO2 then in this? When I'm talking about this complex, where do you lose the CO2? Remember in PDC, you lost the CO2 when you used TPP? That when I showed you this, right? If this was pyruvate, and it was 3 carbons, I lost CO2 here when I used TPP, and then all of a sudden I got TPP. See? Remember that? Yeah, that's what's happening there. It used TPP as well to get rid of carbon dioxide. So you see the analogy here? How honestly you don't even have to work that hard to remember, like to understand these stuff. If you get PVC, just apply the same knowledge here. Okay? Obviously you should probably go back in the slides and make sure your knowledge is right, because that would be really bad if you didn't understand this and then you just totally botched that, okay? So understand this and find the correlation stuff. Make connections, okay? Because as soon as you make connections, you're going to get questions much more correct, okay? Mention that you should probably know the carbons and find them in the citric acid cycle, right? I did say that? Okay, make sure you know that. Okay, I think I'm done, honestly, with this. Yeah. Ending citric acid cycle, that's it. If there's any questions, though, ask me right now, and I can talk about it. Or I'll hold off to the end, so I can do electron transport chain really quickly. Before I get to that, I need to ask you guys, where does the citric acid cycle happen? No.
Oh, matrix?
Mitochondrial matrix. Okay, I just want to say something. Up until glycolysis, okay, when you got that pyruvate, it was made in the cytosol, all right? It was made in the cytosol. But the thing is that to do the citric acid cycle, it needs to go into the mitochondrial matrix. Doing the PDC complex creates acetyl-CoA and transports it into the mitochondrial matrix. That's how you're able to do also citric acid cycle, because of the PDC complex converts pyruvate into acetyl-CoA to get inside. Does that make sense? What I'm saying? So then when we talk about the electron transport chain, what we're saying here is that you have obviously your complexes inside the mitochondria. This is my mitochondria, right? And of course, right? This is called the inner membrane. Okay, this is called the outer membrane. And this is called the inner membrane space. For some reason, people always get confused about where the hydrogens are getting pumped into and stuff like that. So if this is where complex 1, complex 2, complex 3, and complex 4 of the electron transport chain are, then it's pretty easy to tell you that the hydrogens are getting accumulated here, right? So I just want to make that clear to you where you're at when we're talking about electron transport chain. We are in the mitochondria, our complexes are on the inner membrane, and we are pumping hydrogens into the IMS. Just so you know, the IMS already has a lot of hydrogens. Keep that in mind, right? If it has a lot of hydrogens already, then it is pretty hard to push hydrogens into an area that already has a lot of something. And that's where oxidative phosphorylation comes in, specifically the oxidation part. I'm going to show you now. Okay? So, let's do this. This is my membrane, okay? Complex 1, complex 2, complex 3, and then complex 4. Okay, for complex 3, I'll probably have to do Q cycle. Does everyone understand the Q cycle, or would I have to go over it again? If you don't understand, say yes. Just say I need help. I don't care. Yeah. So this is the oxidative part, right? We want to pump hydrogens into the inner membrane space. For starters, this is called the N side, and this is called the P side. Why do you think it's called N side? Why do you think it's called P side? Why do you think?
Proton side.
Yeah, close. It's actually, and basically the same thing, it's because as you pump hydrogens, more hydrogens into this side, into the inner membrane space, it becomes more positive. If it becomes more positive, you just call it the P side. And this side, you're losing hydrogens, it becomes N side, more negative, all right? So, complex 1, she calls it a different name. I call it NADH dehydrogenase complex, and that's what my books told me for other classes. She calls it though the ubiquinone oxidoreductase complex. They're the same thing. I checked it on the internet. I guess you should probably go with what she says, right? But just so you know, in the future, it is usually called NADH dehydrogenase complex, and that's because of this, right? You get all this NADH from citric acid cycle, from all of these places, even glycolysis. What's happening here is NADH is going to come onto complex 1. It's going to give its electrons away. These electrons, guys, are going to start climbing through other electron carriers, like iron-sulfur clusters. And eventually, it's going to be given to ubiquinol. Okay? So what's the purpose of this? Oh yeah, and let me tell you as well. This complex will pump 4 hydrogens across. So what does that mean? I'll explain it. You can take this similar idea, what I'm going to say, and apply it to the rest of the complexes. Except for complex 2, succinate dehydrogenase, you do not pump hydrogens. No hydrogens get pumped in complex 2. That's the only complex, by the way, that does not allow that. But what's happening here is you're getting these electron carriers, okay? When you get these electron carriers, you want to give them from low affinity carriers onto high affinity carriers. High affinity meaning that I eventually want to get to oxygen. You eventually want to get to your last electron acceptor, oxygen. This is the highest affinity. So it makes sense that if electrons are climbing from NADH to sulfur to ubiquinone, right, onto these other electron carriers to eventually get to oxygen, right, that this is the electron transport chain. This is the electron transport chain. You're just transferring electrons from low affinity carriers to higher affinity carriers to get to oxygen. And eventually what oxygen does, it just reduces to water. And I'll explain it in a second, okay? So, you know that iron, sulfur clusters are an electron carrier, okay? But the thing is that with this electron carrier, it is only able to carry one electron at a time. So one electron at a time. The other electron carrier that we have is called cytochrome C. Cytochrome C, though, is actually found on complex 3. But, like anything, it goes from cytochrome C oxidized to cytochrome C reduced. The reason why it's reduced, well, you just put a bunch of electron carriers on it, right? The whole idea is to move electrons onto electron carriers. Cytochrome C, from oxidized to reduced, gets an electron carrier. I'm going to explain it in a second. So, yeah, you see that? Okay. The next one I'm going to talk about is ubiquinone. Ubiquinone is also an electron carrier. It actually looks like this. Q. That's just ubiquinone. But as soon as I add electrons to it, like through the electron transport chain, because remember, you're trying to get to here eventually, so it's going to get electrons on it. It's going to look like Q. So I add 2 electrons into it, but this is the one carrier that also allows for hydrogens. I'll add 2 hydrogens as well. And what do I get? QH2. And this thing is a mobile electron carrier, meaning that it is going to go through the membrane. Okay, it's going to go through the membrane. Now you get complex 1. Does anyone have questions on complex 1? I mean, I think I just I just kind of focused on it so you understand the complex and all of the energy carriers behind it. So now, when we talk about succinate dehydrogenase, we are going from FADH2 to FAD. First of all, succinate dehydrogenase, have you seen that somewhere else? Yeah, you have. It's also in the citric acid cycle. This complex is obviously in the citric acid cycle, but it's found in the electron transport chain. You know that when you're going from succinyl-CoA, this is citric acid cycle, succinyl-CoA to succinate, okay, you have to use succinate dehydrogenase, and you make FADH2. The FADH2 that is made in the citric acid cycle is automatically used in complex 2. You use it automatically, and what's going to happen here is the same idea. It's going to go through these clusters. You're going to transfer these electrons through clusters, right? And eventually what's going to happen? You're going to get to ubiquinone. And it's going to become ubiquinol. All right? This complex does not push any hydrogens. It does not, okay? It doesn't. And honestly, if you think about it, it's part of the citric acid cycle, so for it to have been like moving hydrogens doesn't really make sense. I mean, it just doesn't make sense. Can you think about it like that? That if you have it in the citric acid cycle, why would you be using it as a pump as well? You're not. Instead, it's just moving FADH2 automatically and taking it up. Does that make sense? So now, though, I'm going to talk about complex 3. Complex 3 is really important. Why is it important? Because now you guys have all these electron carriers. I've transferred all of these electrons onto ubiquinol, aka coenzyme Q, right? CoQ. You know those vitamins, CoQ10 and things like that? Yeah, I think it's that, to be honest. So what's happening here is that these QH2s, these ubiquinol, are going to converge into cytochrome 3. Cytochrome 3? Oh my god, complex 3. It's going to accumulate into complex 3. Can anyone tell me what complex 3 is called? The official name for it?
Oxioreductase. Cytochrome C oxioreductase.
Okay. I'm automatically telling you right now the job of this thing. Cytochrome C oxidoreductase. Okay? Keep that in your head, write it down, whatever. I'm going to show you how that works. So at complex 3, you're going to take all these QH2s and you're going to do the Q cycle. Okay? The Q cycle, a lot of people have a hard time with. I won't lie, I also had a hard time with. So I'm going to try my best to explain it to you in a way that it will make sense to you too, because honestly, watching videos, sometimes they're too scientific. I'm not gonna do that, all right? So you've got up to here. I'm gonna show you Q cycle now. Then I erase it. What's happening here is that the Q cycle has two parts to it. And just think of it like this. This complex is gigantic, by the way. It's not just like small little thing. It's gigantic. And it has two parts to it. And now you're going to see why we call it cytochrome oxidoreductase. Why it's called that complex, okay? So, I'll show you part 1 here, and I'm going to show you part 2 here. My recommendation for truly being able to understand the Q cycle is if you can write it down like 5 times and not have to like do anything else. Like if you can write it down cold, then you've got this process down. So let's say we're starting with QH2, ubiquinol. Let's take, for example, all the ubiquinol that maybe came from complex 1, right? Because complex 1 makes ubiquinol. We're going to take this ubiquinol, okay, and we're going to bring it into complex 3. The objective of complex 3, so far you've seen how these electrons are being transferred onto these other carriers, higher affinity carriers. Well, there's another higher affinity carrier than QH2, and it's called cytochrome C. You see it right here. It's on the peripheral part of the membrane, okay? Cytochrome C, and it's oxidized right now. You also see it here too. Okay? So the thing is that you want to take QH2 and you want to take one electron and give it to cytochrome C with oxidase. Yeah, oxidase. The reason why it can only take one electron is because that's just how it works. Cytochrome C oxidase can only take one electron, all right? And you know that QH2 has 2 electrons, right? All right, QH2 has 2 electrons into it. So I'm going to show you how it works and why it's 2 parts. At the end, though, once you give it 1 electron, what do you think it becomes?
Cytochrome C reductase.
Yeah, cytochrome C reductase. It's called cytochrome C reductase because you give it an electron, and then you get cytochrome C reductase, and it's going to go to complex 4. That's why it's called cytochrome C oxidoreductase. It's oxidized right now, but it's going to take an electron from QH2 and become reduced. Then it gives it to complex 4, and we're setting the stage for the final electron acceptor of oxygen. So, let's start. QH2 is going to come into the complex. Oh, and we also have another Q there too. Don't forget, there's a Q here too. I know you're kind of like, well, why do you have a Q there? To be honest, I have a hypothesis. Am I right? I'm not sure, but I'll tell you in the end why. So you have this Q, okay? And I have this QH2 that comes inside. This Q is also going to enter the complex. What's going to happen here is that you know there's 2 electrons in ubiquinol, right? I'm going to give 1 electron because cytochrome C oxidase can only carry 1 electron. That's why I'm giving it only 1. So I'll give it 1 electron. But now where do I put the other electron? Well, I'm just going to give it to Q. I'm going to give my second electron to Q. So what happens here, though, is that as soon as I give this electron to Q, it becomes a radical. What do I get? I get the radical. That's it. I get the radical. I mean, it makes sense. When you give 1 electron to something, you're not giving it 2. 2 is usually like, yeah, you're kind of settled, right? Giving it 1 is usually a radical. When you see like superoxides too, a radical. So I give Q, radical. But now, remember, so I gave 2 electrons away right now. One came here and 1 came here. Now I have to do something with my hydrogens. Well, what I'm gonna do is, I'm gonna pump it. I'm gonna pump it to the membrane. I'm gonna pump it to the P side. Do not forget that this is still a complex in the electron transport chain. The whole objective is to push hydrogens across the membrane. It's to push it. Because at the end of the day, we're going to use that for phosphorylation. And I will explain that once I get there. I'm not going to confuse you right now. I'm going to focus here. This 2 hydrogens going to get pumped out. Okay, great. So now all of a sudden, the QH2 gave 2 hydrogens to the inner membrane space. Okay? Don't forget, this space is getting very, very positive right now because we have other parts that are giving more hydrogens to it, right? So this side is very, very positive, called the P side. This side is getting a little bit more negative, right? You're losing those hydrogens, you're pumping it up. Okay, so once you give this electron to cytochrome C oxidase, it's going to turn, it's going to look like this now. Cytochrome C reductase. That's what it's going to look like. And in fact, this thing just comes off of complex 3. Cytochrome C reductase is known as a mobile electron carrier. When we say mobile, we are saying that this thing is not stuck onto complex 3. What we're saying here is that it's mobile, so it's going to move to the next electron transport complex 4, right? So far, you've seen all these electron carriers within their own complexes, like the sulfur clusters, they were within it. We saw with ubiquinol, though, that it's able to transfer through the membrane, right? Ubiquinol went from complex 1 and complex 2, and it went through the membrane to complex 3. So with cytochrome C reductase, this thing is actually going to move, and then eventually it's going to go to complex 4. Okay? So now what happens here is that this is going to look like Q. Right? 2 hydrogens off, 2 electrons gone. Now you get to the next part. Part 2. Part 2 is going to maybe take another ubiquinol. Maybe this ubiquinol came from complex 2. Let's remember, this one is coming from complex 1 because complex 1 generates ubiquinol, but complex 2 also was making ubiquinol. Do not forget that. So this ubiquinol maybe from complex 2 is coming, and what's going to happen here is the same idea. I'm going to give an electron here. I'm going to pump 2H plus up, okay? But then, don't forget that you had a radical. Remember this radical that was here? So far, I gave 1 electron, right? I pumped 2 hydrogen across. Same idea as what I did here. But I'm still having 1 electron, right? Because there was 2 electrons, 2 hydrogens. 2 hydrogens pumped, 1 electron given. I'm going to give this electron here. And what do I get? Q. But wait, you know what else I'm going to do? I'm going to give it hydrogens. 2 hydrogens. Just wait. I'm going to give this thing 2 hydrogens, obviously because it's from the N side. And I'm going to get QH2. And eventually, let me just explain something real quickly to you. First of all, look at this side and look at that side. You know at the beginning how I said I'm not sure why we have this Q here? I really don't know why. But notice in the very end of the day, you still got the exact same stuff. I personally believe it's because this process is continuous. You're starting with the same reactants and you're ending with the same products, right? So it's continuously going in this cycle. That's my hypothesis of why you're having this Q here, because otherwise I have no idea, right? You should probably check the internet or ask Dr. Erb. But do you guys understand what has happened so far? Right? 1 electron was given to the radical, because the radical was still in part 1. I got Q. I'm going to give it 2 hydrogens to regenerate ubiquinol, because maybe I'm going to do this process again. I don't know. But then I had another QH2, right? QH2 may have came, 1 second. Maybe this QH2 came from somewhere else, right? I gave 1 electron to cytochrome C oxidase, became cytochrome C reductase. And where do you think that goes?
Complex 4.
Yeah, it goes to complex 4 as well. Does that make sense to everybody? Does Q cycle make more sense to you than it did before at least? Yes or no? Say
Yep.
yes. I think so. Does this make sense? Yes.
So, when you raise the H2, do you want us to
I mean, honestly, I would erase it because technically say, so the reason why I erased the H's here is because you did give the hydrogen into the inner membrane space. You did give an electron here, and you gave the electron to radical. Same idea in part 2. I brought another QH2, pumped 2 hydrogens up, gave 1 to cytochrome C oxidase. Remember, I gave it there because that's an electron carrier, becomes reduced. And then of course the radical, I give an electron to it, becomes that. So yeah, I wrote it as Q because that's probably the proper way of writing it.
So the QH2 was actually it was the same thing originally as the QH2 that's there, right?
So this QH2 is maybe from complex 1, right? This QH2 maybe from complex 2 or something. Does that make sense? I don't know where it's coming from. They're from separate areas. Because look, this generated a Q again. It didn't generate QH2. You got this from somewhere else.
Yeah, I know, but like the QH2 that you had inside was represented.
Oh, it was that. Yeah, it was that. Yeah, of course. Are we good? Should I erase it? Can I erase it? Do you have questions? Anything? Are we good?
I have a question.
Yeah.
So for complex 3, is there like 2 separate pieces to it? Like there's 2 little complexes within complex 3, or is it just
No,
one?
I thought about that too.
Okay.
I don't have an answer for you.
That's okay.
I don't.
No
I'm
worries.
sorry. I don't want to give you something wrong.
No, you're fine.
What I thought was is that it was a complex and it had those 2 within the complex and there was a little space between it.
Okay.
Maybe.
I'll look it up.
Yeah, you should look it up. Okay, that's that. Okay, I have to say this because you don't want to get this wrong on your exam. 1 cycle of the Q cycle is going to get you 1 cycle, just 1. 1 cycle of Q is going to only get you 2 cytochrome C reductase. It only gets you 2, okay? And you also only pump 4 hydrogens. Okay? That's just 1 cycle. I told you that eventually these reductases are going to go into complex 4, right? They eventually go to complex 4. There's a problem though. The problem is this. What's the final electron acceptor? Oxygen, right? Oxygen is going to take the electrons, right? Because it's the final one, so it's going to take whatever electrons are left, and it's going to make what? Water, right? It makes water. It reduces to water. It takes all the electrons it has, and then it finally reduces to water. Okay, first of all, cytochrome C reductase carries what? 1 electron, right? And this one also carries 1 electron, right? When you go into complex 4 though, I'm going to do something real quickly. Actually, I'm not going to erase this right now. I'm going to write below it, okay? Let's put it black. Oh, thank you so much. I really appreciate it. Okay. This is just, I'm talking about something else right now that has to do with this. When we talk about complex 4, this is where you get the electron acceptors, right? We're going to go from O2 to water. Don't worry about the coefficient right now, okay? Don't worry about the coefficient. I'm just telling you that this is the last electron acceptor. It's going to finally reduce to water. The thing is that the only way to reduce to water is that you need enough electrons for it, right? You need to get enough electrons in order to reduce there. This thing is only making 2 electrons. It only brings 2 electrons, right? In 1 cycle, you have 2 cytochrome C reductases that hold 2 electrons. In order to fully reduce oxygen into water, it needs 4 electrons. So what am I telling you? I'm telling you that you have to do the Q cycle twice. In total, you need to do it twice. Does that make sense? You look confused. Are you okay? Okay.
So we need to do it twice in order to reduce oxygen to water.
Oxygen needs 4 electrons. But this cycle has to happen twice. Right? So unlike those other cycles, you could do it once and you got whatever you needed. Okay. This one you need 4 to get 4. You need to do it 2 times to get 4 electrons. I'm going to show it in a second. Okay, this brings me to the very last complex and I'll call it a day. Okay? I'll give you one question. If I go through the Q cycle once, how many hydrogens are pumped?
4.
Yeah. If I go through the Q cycle twice, how many hydrogens are pumped?
8.
Okay. If I go through the Q cycle once, how many cytochrome C oxidases do I have? How many reductases do I have?
2.
Yeah, 2. How many? Okay. If I go through the Q cycle twice, how many cytochrome C reductases do I have?
4.
4. Let me just write a cheat sheet right now. You go through it once, you get 2 cytochrome reductases, meaning that each one carries 2 electrons, right? If I go through it twice, I get 4 cytochrome C reductase, meaning that I get 4 electrons, right? Now, when we talk about complex 4, what's happening? Yeah, exactly. They go to O2. Let's see. Sorry, I just want to make sure I have the right image when I'm writing it. Okay, the image that I'm going to draw is on slide 36. There is another slide that has the exact same image. Do not refer to that slide. I'm going to tell you not to refer to it because it's actually wrong. Okay? Slide 36 is the slide that you want to actually look at. Do not confuse yourself with the other slide. I don't know what number that is. I think it might be the one prior to it or after it. Not sure. Cytochrome C reductase. Okay? You need to do it twice because 4 electrons are going to be used to be 4 electrons accepted by O2. Okay? You need 4 electrons to be accepted by O2 in complex 4. So I'm going to draw complex 4 real quickly for you. Remember, I'm going to be bringing 4 cytochrome C reductases. So I'm going to draw it right here. 4 cytochrome C reductases are going to be brought. Okay? I'm going to bring it, and these electrons are going to be dispersed into those copper centers. Remember those copper centers? Those are also electron carriers. They're going to be taking it. All right? I'm just going to make it like F. And obviously there's a heme center here. Do not forget the heme. Star it or something. I don't know if it will come on your exam. There's a heme group in cytochrome C or in complex 4. There's a heme group. The heme group is just there to, I guess, also transfer those electrons. All right? So these electrons are getting transmitted from 4. So 4 electrons are going through, and then I'm going to continue going. It's going to go on to the next one. And then after that, yeah, this is taking me forever. Honestly, this is a terrible picture. I'm going to draw it myself. The O2 is here. I don't know if it should be anywhere different, so I'm not going to draw it here because that's my mistake. I don't want to make everyone get something wrong because of what I did. So I'm trying to correct myself. This oxygen is going to come in, okay? And it's going to give 4 electrons, right? So you're going to get 4 electrons on it, right? Because that's what we've been transferring, 4 electrons so far. After that, what's going to happen here is that it's going to exit, okay? And we're going to give, in addition to the 4 electrons that were given to oxygen, we're going to give it 4 hydrogens. 4 hydrogens. As soon as you are given those 4 hydrogens, you are going to develop water. Notice that it says 2 here. The reason why it's saying 2 is because of this. It is saying that if I have 1 oxygen molecule, and I have 4 electrons with it, and I have 4 H plus with it, right? It's going to make 2H2O. But now, and these 4 electrons came from what? 2 cycles of the Q cycle. If you did 1 cycle of the Q cycle, it would actually be half an oxygen plus 2 electrons plus 2 hydrogens, and that would just get you 1 water. Do you see what I just did? All I'm saying is that to have a fully reduced oxygen, you need the 4 electrons, you need 2 cycles of the Q cycle, okay? If you lose only half an oxygen, you do the cycle once if you do half an oxygen. And that will only get you 1 molecule of water. But the cycle doesn't work like that. It needs to have 2 water molecules. Once you have all this, don't forget 2 hydrogens get pumped out. Okay? No, it's not 2, it's 4. See, there's a drawing there that's wrong, and I looked at that one. It's 4 hydrogens, which makes sense. So 4 hydrogens get pumped out, 4 hydrogens used to make 2 water. Are we good? Yeah? Okay, I'm ready. So once you get all these hydrogens into the inner membrane space, what's gonna happen is you're finally gonna get to the last part of it, the phosphorylation part. The phosphorylation part is literally where you make ATP. So, if for example, so far you've gone through the electron transport chain, you have complex 1, complex 2, complex 3, complex 4, you got all that hydrogen on, there's nothing here right now. Pretend this was a membrane what I just did. So far you've got all these hydrogens, okay, I'm just gonna draw them as plus signs, right? I have all these hydrogens in here, right? And I have finally got to the last electron acceptor. The next thing you're gonna do, you just did a lot of oxidation, right? You just did the oxidation part of it, right? Going from that NADH to NAD+, transfer electrons. We're gonna do phosphorylation. So, what phosphorylation is saying is that it takes a lot of energy to do ADP plus phosphate to make ATP. It takes a lot of energy to do it. So where do you get that energy from? That's the whole objective of why we pump those hydrogens through. When you pump those hydrogens through, okay? When you pump them through, you were putting them into an area that already had hydrogens to begin with, right? When you're going to do the ATP part, it's gonna fall back down its gradient. When it falls back down its gradient, that is where we make ATP, because the energy that is released from going from a high concentration of something to something of an area of low concentration, we can take that energy and put it into making ATP. That is how you do it. Now, I'm going to show you kind of like, I guess, the mechanism part of it. I'm going to draw the ATP synthase, this model real quick, okay? Okay, so what's happening here? We are now at the ATP synthase part, right? We have a lot of hydrogens on this side, right? We have a lot of hydrogens on the P side, and on the N side, not so much. So what we're going to do is that the electron transport chain, everything we've done so far, is going to set up the proton motive force. The proton motive force is these hydrogens coming back inside. And you know what they're going to do? The hydrogens are actually going to come through FO, F naught. You call it F naught, F0, like that. It's going to come through F naught. As soon as it comes through F naught, this is called the gamma stalk. There's a gamma stalk. This gamma stalk is going to start rotating, okay? And on this stalk, we actually have alpha, beta units. You have alpha, beta units on this stalk, multiple ones, right? You have multiple ones. What is going to happen here is these hydrogens are going to come in, the stalk is going to spin. As the stalk is spinning, the alpha beta subunits are also spinning with it inside of each subunit, okay? they are paired together, alpha with beta, alpha with beta, alpha with beta. Each part of it has a different orientation. So when you're spinning it, you're in some sort of orientation. You're either looking like this or you're looking like that. You're looking at a specific type of model, okay? I'm not going to use the words yet, so I can show you. So now you see, right, how it's coming in. It's going to flood through here, okay? This thing is going to rotate, right? It's going to rotate, which evidently causes those alpha beta subunits to rotate. Now, as this is rotating, notice that it's the alpha beta subunits that are taking the ATP phosphate and pushing it to ATP. And that is what I'm talking about when I say orientation. It has different orientations for it. So, let me show you these orientations. We call this the binding model. So the binding model is when we're talking about what rotation we're in, what orientation we're in. So you got this. I'm going to really quickly now draw this orientation, okay? The binding model is what we call it. So if this is going to be a bird's eye view, if I'm looking down the stalk, right, this is what you'll see. Oh, wait. So you can see already that I've kind of linked each one by shape together. These two alpha beta subunits are together, these two alpha beta subunits are together, and these two alpha beta subunits are together. For every alpha subunit, you have a beta subunit. Now, what's going to happen here is that as I'm looking down, so this is a bird's eye view, the stalk is probably right here. This is where the stalk is, right? Alright. That's a gamma, okay? That's where the stalk is if I'm looking down. As I'm looking down, this thing is rotating, right? So this thing is rotating bird's eye view. You see what I'm saying? Right? We're looking down at the gamma stalk. We see this thing as hydrogens are coming in. Gamma stalk is moving. So the alpha beta subunits that are tied to it are also going to rotate with it. Alright? So you see that. Now, in each subunit, we're going to have an orientation. And I'm going to show you for one of them. Okay? So this is alpha beta subunit for the circle part of it, right, when we're looking down. We see that there's an ADP right now, PI, in this subunit when we're just originally starting, right? As soon as you add 3 hydrogens into the F naught, so as soon as I pump 3 hydrogens from the P space, remember because it's falling down its concentration gradient, right? Because it's ATP synthase, it's going to come from the outside, the inner membrane space, it's going to come down. So this is coming from the P side, and it's going to go into the N side. That's going to rotate the gamma stalk. And that's why we see this rotation happening, okay? What's going to happen here is that this ATP-PI is now going to become ATP, right? It's still in this subunit right here, all right? It is going to become ATP. Now, after I get that, this is also still beta, okay? So you get ATP. That's just adding 3 hydrogens in right now. As soon as you add another 3 hydrogens from the P space into the N space, so 3 hydrogens from the outside coming back down, so add another 3 plus P. Okay? This thing is going to move again. And instead, what's going to happen here is that in this beta subunit, it's going to be empty now. Why do you think it's empty? It's empty. There's nothing there. What happened here is that the ATP got released. That's what happened. If you do it again and add another H3 from the outside to the inside again, you restart the process. Maybe it'll be in a different subunit, okay? But notice that it is continuously in beta that we're seeing this in so far, right? I'm just showing you right now in regards to the beta subunit. Do you see how every time you add hydrogens from the outside to the inside, this thing is spinning and you're getting different orientations of it? Every time you add 3, I had ADP PI, all of a sudden I get ATP. I add another 3 hydrogens, this thing is going to rotate again, and I'm going to release the ATP. Okay, that's what's happening here. So let me ask you one question. I'm showing you in this image only one ATP. You have to understand, though, that all of these things are in different spots. So, give you an example. If this right now is an ADP PI, then maybe this subunit right here has, maybe this subunit here has ATP in it, right? And maybe this subunit right here just released ATP, right? So this one has it. This one has the reactants for it. This one makes ATP, and this one releases ATP. Then when you add another 3 hydrogens inside, this thing will make ATP, and maybe this will go back into ADP plus PI, and maybe this one will release ATP, right? This one releases ATP. Then I'll go back here.
Sophia, I think it's the pointy one at the bottom. Shouldn't it be
Yeah.
in the
Yeah.
square?
That's the,
Yeah.
Other than that, I think it's okay.
Yeah. No, you're right. I kind of noticed that myself. I was like, why does that look? Oops. Yeah. So this one should actually be. Yeah. This one should be the one releasing. My bad. And then this one should be the. No,
Are we
so
still
what the hell? This one is
going the other way?
releasing. Yeah. This one's releasing.
Yeah.
So this one releases ATP, sorry. And then this is the one that is going to be making ADP plus PI, yep. I'm sorry. And then this one would actually be okay, that makes more sense. Does this make sense? I know I have to stop it for a second. Do I need to re-explain it, or you think you got it? I feel bad though because I've recorded this. It does make sense though, right? The idea that I showed you in the beginning the first part of it, but notice that these subunits still have something happening in them. So on the exam, if I ask you, how many hydrogens are put into the full cycle, how many hydrogens would be put in? How many hydrogens put in from the top air, from the inner membrane space, flooding back through ATP synthase?
9.
9, right? Yeah, 9. Because 3, 9, right? You added 3 each time, okay? To spin this thing. Each time you spin it, notice that every spin for each respective subunit, right? Because this subunit is different from that subunit, which is different from this subunit. Because this is a triangle, that's a square, and this is a circle kind of one, right? Each one is releasing ATP. I get released ATP from the triangle, I get released ATP from the square side of it, and I get released ATP from the circle. So if I ask you how much ATP is made from one full rotation, how many would you say?
3.
3, right? You get 3. If I ask you a third of a cycle, what do you say?
1 ATP.
If I say 3, if I tell you 1/3 of a cycle, that's just like this. I only add 3, and you only make 1 ATP in a third of a cycle, right? Because there's 3, so 3 ATP, 9 hydrogens for the full thing. If I look at only a third of the cycle, 3 hydrogens added and 1 ATP made. That's it. I'm sorry about that picture stuff. I was struggling, but we're good. Okay, I call it a night.